Okay, hope this is working. This should be the only time that I do this video by myself. I'm gonna try and keep it brief and keep it interesting. So I'm just gonna start with the question one. So I don't have it all up on the computer, but I have things written. I can show drawings. Question one, the one about blood types, the ABO blood group in humans sorts independently from the rhesus RH blood group trait and the MN blood group trait. The three alleles are possible, the A, B, and I, and for the ABO locus, the two alleles are, and a dominant allele producing a RH plus and R, a recessive allele for R H negative are found on the RH locus, and codominant alleles M and N occur on the MN locus. Each gene is autosomal. So just going through these questions. A child with a blood type O, RH, and an M is born to a woman who has blood types O, RH negative, and MN, and a man who has blood types A, RH plus, and an M. Determine a possible genotype for parent and for the child. So for the mother, in order for the child to have an O blood type, he has to have homozygous recessive. So the parents are going to have to either be heterozygous. The mother is O, so she's going to be homozygous recessive as well. The father is going to need to be homozygous or heterozygous, and have, or in other words, have an IA and a lowercase I in order to pass that on to the child, if you even have a possibility that the child, because if he was homozygous, then just that IA will pass on and it would dominate over the lowercase I, or the recessive allele. So the mother, and then the child also is in RH negative, so it's kind of the same thing. He needs to have two lowercase r's. So the mother, who is RH negative, already has two lowercase r's. The father is RH plus. He's going to need to be heterozygous. So and then the M, the child is M, or just MN, or just an MM, and the mother is an MN, and the father is an M. So there's only those possible genotypes for them. So what I got was the mother has a lowercase I, lowercase r, and an MN for your genotype. The father is, or I should explain it better, is heterozygous A blood type, so IA with a lowercase I, heterozygous for the RH plus blood type, so capital R, lowercase r, and an MN. And the child is homozygous recessive, or O blood type with a little I. He's RH negative with a little r, and he's M with that. It has to be MM. So these are the only possible genotypes, since there's only one genotype for the O blood type, the M, the MN, and the RH negative. The father would have to be heterozygous in order to father a child with a homozygous recessive genotype. So then with B and C and D, I did a lot of Punnett squares. So what portion of a child born to a man with a genotype IB, R, or homozygous, or heterozygous R, and NN, and a woman with a AI, or a heterozygous A blood type with a RH plus, but it's heterozygous, and an NN will have a blood type B RH negative and MN. So what I had to do there, so I was going to look backwards. But what I just did was I basically just followed exactly what it said for the man's genotype and the woman's genotype. I just did a Punnett square for their blood type, for their RH blood type or blood group, and for their MN blood type, and just showed that 1/4 of their sorry, it's totally backwards, but what it's written here is 1/4 of their offspring will be of blood type B, 1/4 will are RH negative, and 1/2 will be MN. And just like a split tree diagram, you have to multiply those probabilities. So if 1/4 of their offspring will have a blood type B, 1/4 of that 1/4 will have RH negative, and 1/2 of that 1/4 of 1/4 would be MN. So 1/32 would be B, RH negative, MN. So I did the same thing with the next one. So it gives the man's genotypes, and then it says what portion of them will have those traits. And I went through and did it again and multiplied them through, and it was 3/64 would be A, RH plus, and M. And then it says the last one is a little different. It says a man with a blood type, basically gives a man's blood type, says he could not be the father of a child with a blood type O, negative, MN, and the mother of the child has a blood type A, RH plus, and MN. Is this man correct? Explain. So he's basically saying, well, I have blood type B, and the mother, and I can have a kid that's blood type O. But if he is heterozygous, so if he had a B, so we don't know what his genotype is, just his phenotype. So if he was heterozygous with a B, or IB with a little I, and the mother was also heterozygous, then they could possibly have a child. 1/4 of their children would be would have the blood type O, and then 1/4 of that would have if they were heterozygous for their respective plus or negative or minus blood types, they will also have a they'll have 1/4 of those will be negative, and then 1/2 of them, if they are heterozygous, or well, if you just follow those blood types of MN and M, or it's just NN and then plus MN. Kind of shows it right there. So 1/4 or 1/2 of them would be NN, and so 1/32 would be of their children would be O negative MN. So he was wrong. If he's that 1/32. Okay, so that's question 1. Question 2. It's really long to kind of explain everything, but in class we learned that the comb shape in chickens is determined by the alleles of 2 independent assorting genes: R, dominant R, or big R, little r, big P, little p. So it kind of explains the same things that we did in class about how they are, what each combination means. And so I went through, and it just is A is just like just giving these different genotype ratios, or these phenotype ratios. You should figure out the genotype. So it's just basically the way to solve this is to kind of play with it, I guess. So for each one of these ratios, I just went through and so I can see it's backwards, but did these different genotypes, which corresponded to the phenotypes that we were talking about. Actually, no, these are just the genotypes, and I'm trying to kind of get a phenotype ratio that is the same as A, B, and C. So I just kind of knew that I needed to have kind of 2 different, I don't know, basically you just have to play with it and try different combinations just make up a certain genotype, make a Punnett square of it, and you'll kind of see the ratios that are showing up, and you'll see, okay, well, I need to change this little r for a big R in order to get a walnut in there. So if I want a whole row or a whole column of walnuts, I got to make sure that there's a uppercase R in there and an uppercase P. Those are the two that I'm looking at. So as long as those both are sorted down or in there, okay, I'm going to get one that is walnut. This one is going to be rose, this is going to be P, and then this is going to be single. So that's kind of how I followed it on that. And then I just had to change it up and find which ratios would give a 75% rose and 25% single for the B. Moving on, which ones would give a 50-50 split for rose and single. Sorry that it's backwards. And I just had to change it up. So see, there's only one there's only one dominant R or dominant allele in any of this, and that was what passed down in at least two of these rows, and that was what caused a rose. But then having these recessive ones, that was a single. That's what I was looking for. And so same for D. I wanted a 3 or a 75, 25, or 3-1 split. And so I kind of had to change up with the different alleles. So you just gotta buckle down and write them out, or else you're not gonna get them. So that's and yeah, it proves. Sorry, I don't have it all perfectly written out. The next one is just for number. I'm out of line. For number 3. There is a Palomino horse has a golden yellow coat, chestnut horse has a brown coat, and a Cremello horse has a coat that is almost white. A series of crosses between the 3 different types of horses produce the following offspring, and it gives you this list of what happens when you mix certain colored horses. So just by looking at it, you'll see that the only time that there's a split is when there's a Palomino in there. So, and I'm not 100% sure if this is more related to kind of the skin color stuff that we learned about, and that when there's more dominant alleles, the more dominant alleles there are, the darker the skin color. So that was for humans. So if there's like 3 shades, which Cremello being the lightest, Palomino being the middle lightest, the middle one, and the chestnut being the darkest, that is a possibility. But if you see, Palomino would be the heterozygous one. It would have I don't want to say dominant, because when chestnut and Cremello mix, it's not all chestnut or all dominant. It doesn't dominate over it. It's like co-dominance, or it's controlled by more than one allele. So just really quick, we did Palomino, for example. I'm just going to do T's. So let's say that a Palomino has a big T, little t, not meaning that the T is dominant over it, but just the more mix will cause a Palomino. This would show that oops. So that would cause, sorry, they are backwards, but if the Palomino is a split of a big T, little t, and that's also a Palomino, big T, little t, and then we want to say that the two T's, the uppercase T's are the chestnut, and the two lowercase t's are the Carmelo. That would make the same ratio of a 13 Palomino, 6 chestnut, and 5 Carmelos, give or take, because you can't make 25% of that into an actual number if with these ratios that we have, but it's very close to half of them are Palomino or mixed, and then 1/4 is chestnut, 1/4 is Carmelo. So then when you mix a chestnut with a chestnut, you're going to get all chestnuts because they're big T. There's no other alleles in there. And the Carmelos are going to be mixed, and they give the exact same. They're all Carmelos because they're lowercase t's, if that's the allele that we're choosing to use. A Palomino with a chestnut, so a mixed with the chestnut would be a big T, would make the same ratio with a big T. I'm just looking on this side. I don't know how to write backwards yet, sorry. So we got a big T, so that's chestnut, and that's Palomino. And they had said when a Palomino mixes with a chestnut, it gets about a 50-50 split, or 8 to 9, which is about the same. And it does the exact same thing with a Palomino to a Carmelo. You're going to have a lowercase t, and it's going to be 50-50 split. So those are the possible genotypes. A Palomino would be a mix of an uppercase, lowercase, or possibly 2 different letters, whatever you want to use. Chestnut would be 2 of the same, but one of those would correspond to the Palomino, and the other one that corresponds to the Palomino would be the 2 of the same in the Carmelo horses. That makes sense. Okay. I'm late. So, moving on to number 4. This one, to answer these questions, you just have to do the reading. So that's the only trick to it. So I'll just read through it and indicate which is the fill-in-the-blank word. So the enzyme encoded for IA and IB alleles is called, so for the blood types A and B, so when the enzymes are glycosyl transferase. For if a person has a B blood, this enzyme adds the sugar galactose to the surface carbohydrate on red blood cells. Overdominance is a genetic phenomenon where heterozygotes have a reproductive advantage over the 2 homozygous classes. What's their example from the reading was the sickle cell anemia. So when it was a mix, it did not, or as heterozygous, it didn't have the sickle cell shape, and didn't have sickle cell anemia, but it was also resistant to malaria. So win-win in that situation. Incomplete penetrance is a genetic phenomenon in which an individual does not express the phenotype specified by their genotype, and in the reading that's the one about the extra digits or the extra fingers or toes. The disease phenylketonuria in humans can be alleviated by modifying a person's diet. And that's from the reading. Mannix traits display dominant inheritance, so that's the dominant allele, while a 2:1 phenotype ratio among the offspring of a monohybrid cross suggests lethal inheritance. So the one that ends up with the big M dies in that example. So the one about the shells coiling in water snails, we only need to look at the females because that's the maternal inheritance. This species shows maternal inheritance. So it says that that 30 have the DD genotype, 30% have the DD, or the heterozygous or homozygous dominant, and for the right-handedness, and 30% are heterozygous, but they're also for the right-handedness, and 40% are homozygous recessive for left-handed. And then it selects about the popular, it's like 50-50 for the males, but it's going to be a 60% dextral, or 50% of them are going to get, or 60, sorry, not 50, 60% will be dextral, because 60% of them, that 30 and 30, will be passing on that dominant D allele, which will cause them to be dextral from the maternal, the nursing cells, while during oogenesis. 40% will be synstral, for that same reason, because 40% have that recessiveness, and the mothers are only going to pass on that little d. Same thing in H. This question considers the leaf pigmentation traits in the 4 o'clock plant and jalapa. So 40% of the female plants are green, while 60% of the female plants are white. So it'll be the same thing in their offspring. It doesn't matter what the males are. So 40% will be green, 60% will be white. So on to question 5. In this one, you just gotta kind of play with it as well. So it tells you 1, 2, 3, 4 different colors in guinea pigs, and the black is dominant over sepia, cream, and white. Sepia is dominant over cream and white, and cream is only dominant over white, and white has to be homozygous, or else it's not going to show up. So I just kind of had to play with it. So if a black homozygous, sorry, black. So a black guinea pig that has a B and an S, so it's heterozygous for black and sepia, will be black. And if a homozygous sepia, if they mate, then half of them will receive that B, that B, or that B end up being black. The other half will be sepia. So there needs to be sepia in both of them for that to pass on. So it would be 50% sepia, 50% or 50% black, 37% sepia. And so with the other ones, you just kind of have to do the same thing. If you do a 2 heterozygous, you're going to get for the same 2 traits, like in B, you're going to end up with a 75 or 3-1 split. That's what we're looking for, 75-25. So both of them have to be sepia, but they also have to carry that heterozygous for cream, and it passes on. If you can see that a C moves down, but the rest have S's in them. It's kind of the same with black. If you want 50%, are going to get that black because it's the most dominant. So one of them needs to be black, and it's going to dominate this column. The white, we just want it to be covered up by sepia, so this one gets covered up, and then white ends up getting it. So if you had a black one that also has a white allele in it, and a sepia that also has a white, in that far bottom corner, you're going to get 2 W's together, making a white guinea pig. So then on the last one, you want another 75-25 split. You got to get 2 heterozygous for the same 2 alleles. In this case, black and white, which will cause a homozygous for black, heterozygous for black and white, and then the last one will be white or homozygous white. So that one will be white. The rest will be black just because it's dominant over that white. So that's basically how you do those ones. You just kind of have to play with them, try different ways, but those are good rules of thumb. 2, like a black and white, and a black and white is going to make a 75-25 split. A SC is going to make a 75-25 split. Basically, that's how you do it. Thank you.